features of DNA replication. So, we know about the general features of DNA replication, and there's some vocabulary terms associated with it, such as bidirectional and semi-discontinuous. Make sure you know what each one means. Usually, and you can go look at the SI videos for more detail, but a very quick explanation of this is bidirectional means that you can open up DNA at one spot and replicate it in both the left and right direction. That is what bidirectional DNA replication means. What semi-discontinuous means, and that's better illustrated with this diagram here, is that because DNA is anti-parallel, you create two separate template strands. One can be synthesized in a continuous manner. They call that the leading strand of DNA. And the other one is made discontinuously. They call that one the lagging strand of DNA. When you put these two terms together, we come up with that overall term called semi-discontinuous. One is made continuously, the other one is made discontinuously. So, one common question that's usually asked about this is you'll be given a picture like this. All right, and you might have, A, B, C, D, and they might ask you which things on here are the leading strand, which things on here are the lagging strand of DNA. Okay, and the way to figure this out, if you can choose to memorize it, that's fine. I personally have to kind of draw out some stuff to figure out which is going to be the leading strand, which is going to be the lagging strand. The main principle is the following. If DNA is made in a 5' to 3' direction, what direction will polymerases read the template in? 3 to 5, exactly. So, let's look at each corner here of this diagram. In this diagram up here, we have a replication fork that is going to go to the left. So, on the top strand of DNA, this is the 3' end of it, and that's the 5' end of it. It is going to read this template strand in the 3' to 5' direction. Whenever the direction DNA reads a template strand is in the same direction as the replication fork, what term do we use to describe that strand? Leading strand, exactly. Just to fully illustrate this, let's look at the other strand. This is the 3' end of that, and this is the 5' end of it. And notice that it would want to read the bottom strand going from left to right, which is the opposite direction as the direction the replication fork is moving in. When they are going in opposite directions, we use the phrase lagging strand. All right. After you figure out half of this, you can very quickly figure out the other half. If A was a leading strand, what's the other leading strand? Yep, the opposite side, which is D. And that means this one is the lagging strand. So you don't have to draw the other side. You only need half of it. Yes?
So it's basically based off the reading, not
yep.
the making. Okay.
The direction that the enzyme wants to read the template in.
So 3 to 5.
Yep, it reads it. Okay. Polymerases read template in that direction, so be very careful with that, yes. All right. They may ask it in slightly different ways, such as, where on here are you going to find Okazaki fragments? And that is another way of asking you which one is the lagging strand, right? Because the lagging strand is made discontinuously with Okazaki fragments, yes?
Isn't it technically right from 3 to 5, but it's also made then 5 to 3 on the new strand?
Yes.
So you can think about it that way too. It's not
Whichever way you prefer doing, if you want to draw the new strand of DNA, that's fine too. It will give you the same answer either way. Excuse me. All right, so Okazaki fragments are found in the lagging strand. Where are you going to find DNA polymerase I? On the lagging strands also, and that's because, and we'll get to it in a little bit, DNA polymerase I is involved in what named process? Nick translation, right? And we'll go over that in a little bit. Okay, back to this. Okay, so I'll do it in this order. I'll do DNA polymerase III first. Okay. In DNA replication, there's a bunch of enzymes involved. Helicase, the function of helicase is to unwind DNA. We have a set of enzymes that fix supercoiling called topoisomerases or gyrases. I'm not writing this stuff down because we've gone over it a million times, but what enzyme makes primers? Primates, right? Really easy stuff. The important part, the really important part, are the DNA polymerases because they are the things actually synthesizing DNA. DNA polymerase III, the job of DNA polymerase III is to make most of the DNA in a prokaryote. Oh yeah, I should mention, this information here is for prokaryotes, okay? And so, because of that, I am going to make a page for prokaryotic DNA replication. Prokaryotic DNA replication. All right. Prokaryotic DNA replication. What do we have here? We have prokaryotic DNA replication is the replication of circular DNA. And does anyone know how many origins of replication do you need to replicate circular DNA? Just one origin of replication, sometimes called one ORI-C site. And we've drawn what the ORI-C site will look like. The fact that it has two replication forks going in opposite directions. We drew that earlier. And it involves DNA polymerase III and DNA polymerase I. DNA polymerase III is the main one. It makes most of your DNA. It synthesizes everything on the leading strand. And it's the one that makes the Okazaki fragments on the lagging strand. How does it do that? How does it also synthesize DNA on the lagging strand? It does so by forming a loop on the lagging strand, which is in one of those pictures that you see in chapter 28. But it forms a loop on the lagging strand in order to synthesize the fragments of DNA. DNA polymerase III is a multi-subunit enzyme. It's made of an alpha subunit, a beta subunit, epsilon, and theta subunits. Okay? The alpha subunit is your 5' to 3' polymerase. Is your 5' to 3' polymerase. Whenever you see that phrase in DNA polymerase III or any other enzyme, it means that's the thing that's actually synthesizing DNA. That's the thing that is replicating DNA. So the alpha subunit has that particular function. The beta subunit has a nickname called the sliding clamp. So what the beta subunit does is that it keeps DNA polymerase III bound to the template strand of DNA. In doing so, it forces DNA polymerase III to synthesize tons and tons of DNA without stopping for a break. And that literally is what the beta subunit's function is. In contrast, DNA polymerase I does not have a beta sliding clamp, and so DNA polymerase I doesn't synthesize as many nucleotides in a row. DNA polymerase III, though, is able to have what is called enormous processivity because of the beta sliding clamp. The epsilon subunit, there's a phrase here, 3' to 5' exonuclease. This is your editor function in terms of DNA replication. Yes?
So alpha is a proofreader?
Yes. So after I'll explain that after I do the editor thing. So 3' to 5' exonuclease, basically what this is functional-wise is it removes nucleotides. And the only time you want that to happen is if you made a mistake. So if this DNA polymerase is adding nucleotides one at a time as a complementary to a template strand of DNA, if it makes a mistake, the one that actually detects the mistake is the 5' to 3' polymerase. This is your proofreader. And how does it do that? Well, remember, G's and C's will have 3 hydrogen bonds, A's and T's will have 2 hydrogen bonds. So it knows most of the time when it makes a mistake if the number of hydrogen bonds don't match up to each other. So when it detects this mistake, the editor will immediately remove that nucleotide. And what you end up creating where this happens is on the shorter strand of DNA. You remove the most recent nucleotide that was added, the wrong nucleotide. You remove it using this activity, and you create a 3' OH. You leave behind a 3' OH because you just removed the nucleotide from there. So what's left behind is a 3' OH. You are now able to add the correct nucleotide to this position. So it's going to try again and add another nucleotide to this spot here. This spot here, we know it's a 3' OH. There's some other terms sometimes associated with this. Does anyone want to know if this is the anyone can tell me, is this the overhanging or recessed strand of DNA? It is recessed. It is shorter than the other one, so that's why it is called the recessed strand of DNA. So this is the recessed 3' OH. All right.
Does pole 3 usually do this, or does pole 1 usually?
And pole 1 does have the capability of doing this also, which we'll move on to next. So these are the functions of DNA polymerase III. It synthesizes DNA using its 5' to 3' polymerase, and sometimes when it makes a mistake, it'll use this to remove the mistake and it'll try again. You can contrast this with DNA polymerase I, which is one subunit. That's very important. DNA polymerase III has multiple subunits. We only care about 3 of them. DNA polymerase I is just one subunit with 3 functions or 3 domains. Or active sites also works here. Activities, active sites, it all means the same thing. Active sites are where things will go to. All right, what those 3 functions are, we know 2 of them already. 5' to 3' polymerase. Yes, question?
What's the difference between a subunit and a fragment?
A subunit is more of like an intentionally made piece of something. A fragment, and again, this is just, I guess, word definitions. The fragment is when you purposefully cut DNA into pieces, so it isn't naturally made. A subunit is just a fully formed part of a protein that was made naturally. A fragment is something you artificially made. That's sort of what it is. And you're probably talking about the clean-off fragment stuff, right? Yeah, so I'll get to that later. So, one subunit with 3 activities, 2 of which we already know. And they're the same functions here. 5' to 3' polymerase, it replicates DNA. 3' to 5' exonuclease, it edits errors. And brand new function here, this one, called 5' to 3' exonuclease. This activity is only on DNA polymerase I because this thing, its job is to remove primers. Because in DNA replication, primers are made of RNA, not DNA, so that's bad. So if you want to finish DNA replication, all those RNA primers need to be removed, and you need to replace them with actual DNA, which is what the job of DNA polymerase I is in terms of prokaryotic DNA replication. So remove the primers, replace them with DNA. So that is what this function is. This is DNA polymerase I. We talked about DNA polymerase III. DNA polymerase I is a component of the process we talked about earlier called a Nick translation, which only occurs on what? The leading or lagging strand? The lagging strand. All right, it only occurs on the lagging strand because the lagging strand has tons of primers on it. Every single Okazaki fragment has a primer there. All right. Technically, there's one other enzyme necessary for Nick translation. Anyone know the name of that? Ligase, exactly. DNA ligase. DNA ligase's job is to finish sealing the nick between 2 Okazaki fragments by forming the sugar, by fusing together the sugar phosphate backbone. Between the 2 Okazaki fragments. All right. 1 second. Let me flip back here. Make sure I missed anything. other prokaryotic specific stuff. Because prokaryotic DNA is circular, we mentioned earlier you only need one origin of replication. And because DNA replication is bidirectional, you create two replication forks in this circular DNA. What eventually happens is these two replication forks will meet each other at the other end of this DNA, and there's a special name for this site called the TER locus. Exactly, the TER locus, which stands for termination locus. It's the spot on prokaryotic DNA where DNA replication ends, and that's where the two replication forks will meet. There's some other information with the TER locus, such as it turns out there is a consensus sequence there. You don't have to memorize the consensus sequence, but there is a consensus sequence there. And at that consensus sequence, there's a special protein that binds to it. That protein is called the TUS protein, exactly.
So the TUS looks for the consensus.
Exactly, yep. And you might notice here, and you technically learn about it in chapter 29, that consensus sequences are for DNA binding proteins. The TUS protein is a DNA binding protein, but it's not like a transcription factor or activator of something. What function does the TUS protein have? Contrahelicase, exactly. And what that means is, you know these two replication forks, they each have a helicase in order to open up DNA to continue replicating. When they reach this other end, there's a TUS protein waiting for it, and it will shut off those helicases with its contrahelicase activity. All right? Let's see. All right, so another sheet of paper we're going to need because we're now going to talk about eukaryotic transcription. Did I say transcription? Excuse me, eukaryotic replication. And you might notice that there's less information that's necessary when you compare it to prokaryotic replication. Eukaryotic replication, eukaryotic DNA is linear and is a lot longer than prokaryotic DNA. Prokaryotic DNA, there's less bases in it. So eukaryotic DNA, because it's linear and because there's way more, you need multiple origins of replication.
Does that make eukaryotic replication faster?
That is a good question. In general, it is, but not because of the multiple origins of replication. That is a more complex thing that we don't need to go over for this class. So don't worry about that. But it does try to be as fast as prokaryotic replication by making multiple origins of replication. Each origin of replication, of course, will create two replication forks. And at each of those replication forks, you have DNA polymerases replicating DNA. So the important part for eukaryotic replication are your different DNA polymerases. DNA polymerase alpha and the rest that I never remember. So that is all on here for you guys. I'm not going to bother breaking down each one. Make sure you do know every single one and the differences between them, the activities that each one has. So DNA polymerase alpha is special because it is able to make its own RNA primers. DNA polymerase beta is similar, except it's mostly involved in repairing. DNA polymerase gamma is really interesting because it only replicates DNA in the mitochondria and chloroplasts, even though chloroplast isn't listed here. And that's unique because eukaryotes have separate DNA in mitochondria and chloroplasts. DNA polymerase delta, and brand new information to me, DNA polymerase epsilon are the two forms of eukaryotic DNA polymerases that make most of the DNA in eukaryotes. So make sure you know what's listed here. DNA polymerase delta is responsible for the lagging strand. DNA polymerase epsilon is responsible for the leading strand of DNA. Yes?
Casey said that they can do both when I asked him. So should we
That
just
each
memorize
one
the
can
lag-
do both, or does he
Yeah.
mean that both of these do
The,
that?
so delta can do both lagging and leading.
Yes, that's what I learned.
So we should
But
learn
for
how
this
to-
class, go with what's listed here. Yes, I learned that DNA polymerase delta actually replicates both strands of DNA. But for the purposes of the exam, we go by what information he has given us on the slides.
But Dr. Casey said that in class today, because we were going over some questions, and he said that if that question's going to be on there, he changed
He would change, so I know what he means. I won't say one way or the other, but go by what the slides say, all right? And in part, and I understand why, because they used to ask it in that DNA polymerase delta is involved in replicating both strands of DNA, but he did want to give you guys the most up-to-date information. You do need to know this thing, that pCNA is a special subunit very similar to the beta sliding clamp that helps DNA polymerases in eukaryotes have enormous processivity. Not necessarily unlimited, but definitely increases the processivity of these DNA polymerases. All right. Very important, telomerases, okay? This is a feature of eukaryotic DNA replication. So if you're following along by separating your notes into different sections, telomerases are telomeres are a eukaryotic specific thing. The reason why is eukaryotic DNA is linear, and because it's linear, we reach a problem when we reach the very end of your template DNA. So on the lagging strand of DNA, remember that we had to make the lagging strand a little bit at a time in terms of Okazaki fragments. We would need to attach a primer every single time we want to make an Okazaki fragment, which is usually fine, except at the very end of DNA, we are unable to attach that very last primer on the lagging strand because there's not enough template DNA to attach the primer to. So, in a lot of cells, they are unable to fix this issue, so they just give up DNA replication at the very end. The end result is that every time that cell replicates, the telomeres, which is the end of DNA, shorten a little bit. And the more and more times it replicates, the DNA will get shorter and shorter until it reaches a term called the Hayflick limit, which means DNA is short to the point that it loses the ability to replicate itself again. It doesn't have enough information to make the enzymes to replicate itself again. It has reached the Hayflick limit. Now, there are special categories of cells that can fix this issue on the lagging strand of your telomere. Those cells will have active telomerase. So telomerase, here's how telomerase works. It's a special enzyme called a reverse transcriptase that works by extending the template lagging strand, okay? By just adding a bunch of bases here in some random pattern that doesn't matter. The telomeres, you don't need to be very specific with the patterns of DNA at the telomeres because you don't have important genes usually at the telomeres. So telomerase extends the template strand, the template lagging strand, so that now there's plenty of room to attach a primer to it. Yes?
Does it use RNA bases to do that?
This one actually is DNA bases, and you can remember that by looking at the activity of the telomerase and that it is a reverse transcriptase. So it uses an RNA template to just make some pattern of DNA bases here, and now that the template strand is sufficiently long enough, you can attach a primer to it, and you can finish replicating the lagging strand. In fact, the specific enzyme that will finish the replicating on the lagging strand at the telomere is DNA polymerase alpha, okay? Because one reason why is DNA polymerase alpha is able to make its own primers, so it can finish off the lagging strand at the telomere, okay? The cells that are able to do this are ones that replicate often and very quickly. That's listed here. Embryonic cells, the ones that are trying to replicate very quickly to form a complex organism. Immune system cells, so when there's some type of immune response, you want those to proliferate quickly. Sex cells, so your germline cells need to be synthesized quickly. Cancer cells is the bad type. Those will replicate over and over again without dying because the telomeres aren't shortening. So all of these have active telomerase. So I use the phrase active telomerase because technically every cell in the body has the gene for telomerase, but only cells that replicate very quickly have the active version of telomerase, okay? So if they ask for which cells have the gene for telomerase, the answer is all cells. But which cells have active telomerase is only the ones that replicate very quickly. All right. Telomerase, we talked about others, PCR, we talked about PCR. Reverse transcriptase. We went over the basics, like what reverse transcriptase does is it takes an RNA template and turns it into DNA. The one example they give in detail is certain viruses have reverse transcriptase. Certain RNA viruses have reverse transcriptase to try to incorporate their genetic information into a host's own DNA. The way it does so is that this RNA virus usually hacks the host's own tRNA as a primer for reverse transcriptase. Then it will reverse transcribe the RNA into DNA and then insert that DNA into the target genome. This is one of the areas of your notes where they talk about HIV and why it's so hard to treat, so make sure you know that. Everywhere in the notes where they mention HIV, review that because there may be one or two questions asking about the features of HIV and why it is hard to treat. Yes?
So why is it hard to treat?
A bunch of different examples. The one example that I always remember off the top of my head, there's others you're going to have to look at the notes for. The one that I remember is that reverse transcriptase does not have a proofreading function. So when it is reverse transcribing RNA into DNA, it makes mistakes. But however, these mistakes aren't serious enough to actually kill the HIV virus. They're just enough to make proteins that are different enough that we can't design drugs to. So that's why it's one reason why we can't treat HIV very easily. Although for some reason, I haven't followed the news that closely, but I know there's some HIV treatment starting to become developed right now, but you don't need to know that for this exam, obviously. Let's see. The way, again, reverse transcriptase works, it's 3 steps based off these 3 activities you see here. We talked about this first one a bunch, RNA-directed DNA polymerase. That means you take an RNA template and you make DNA out of it. RNase hydrolase activity, well, after you made your DNA from the RNA, you've created an RNA-DNA hybrid. So this thing will separate those two from each other. Now that you have your newly synthesized DNA, your single-stranded DNA that was made from your reverse transcription activity, you use DNA-directed DNA polymerase to replicate that single-stranded DNA into a bunch of copies of that DNA strand. So these 3 activities will be found in reverse transcriptase. Yes? Well,
No, you go.
okay, yeah, go
Does
ahead.
that mean that's a second kind of reverse transcriptase, RT2?
Yeah.
Oh, no, this is just unless Dr. Hayes goes over something that specifically, and I haven't heard of that. I think this is just the second slide in regards to reverse transcriptase. Okay. Yes.
Reverse transcription, is that also like found in prokaryotes and eukaryotes, like on both?
I do believe some prokaryotes have reverse transcriptase, viruses have reverse transcriptase, and eukaryotes have reverse transcriptase. So you can find it everywhere. And other questions? All right. cDNA. All right. So, cDNA would kind of be eukaryotic transcription, but it's like a special topic, so I'm going to keep this as its own separate thing. You will need to know about cDNA for exam 3. It is a eukaryotic thing. Let me go over the concept and you'll see why it's kind of hard to classify this. cDNA stands for complementary DNA. Alright? It's a special type of DNA that's kind of like the DNA version of mRNA. Here's what it means. In eukaryotes, remember eukaryotic genes, eukaryotic mRNA is monocistronic, right? So, one of the features of monocistronic genes is that there are, if we have a gene here, there are introns and exons. Make sure this is still recording. Yep, good. So there are introns and exons on this mRNA. In eukaryotes, in order to transcribe this, to fully transcribe this, we splice out the introns and we have our exons. So this is what mRNA looks like in eukaryotes, is only the exons, okay? Now, what if we want to trick a prokaryote into replicating, into synthesizing DNA from a eukaryote? What if we want to do that? Or what if we want to analyze the genome from an mRNA from a eukaryote? Well, we can't just cut out this gene. We can't just cut out the DNA in a eukaryote and insert it into a prokaryote, because the prokaryote doesn't understand introns. It will synthesize these parts of DNA as if it's normal. So what we figured out how to do is we take mRNA, eukaryotic mRNA, and we use reverse transcriptase to create cDNA. So cDNA is called complementary DNA. It is reverse transcribed mRNA in eukaryotes. So we artificially create this. So we artificially create cDNA. cDNA is the DNA version of finished mRNA in eukaryotes. It is special DNA without any introns in it. With this, we can modify it in a couple ways. And I know this is kind of annoying, but it kind of blends in with chapter 30 a bit. But I'll give you the information for context. This cDNA, if we want to trick a prokaryote into synthesizing this gene for us, we have to add one modification to it. We have to add something called a Shine-Dalgarno sequence. That's unfortunately more of a chapter 30 topic, but you do kind of need to recognize this term in case you see it in reference to inserting cDNA into a prokaryote. We need to have the information for a Shine-Dalgarno sequence on cDNA, because a prokaryote will not translate mRNA made from this unless it has a Shine-Dalgarno sequence. So we have to modify it in that fashion. Yes.
So is cDNA mRNA, or is it still like DNA?
cDNA is still DNA, but we're able to use recombinase cloning to clone this fragment of cDNA into a prokaryote to trick it into transcribing and translating it.
So you don't exactly use reverse transcription.
You use it to make the cDNA.
But then wouldn't it make RNA? Never.
Yeah, reverse transcriptase will make the cDNA, and then you insert it into whatever prokaryote you want. And for the final exam, you will be asked about recombinase cloning in detail. So that is the next step here, is that recombinase cloning procedure. All right. Ah, yes, my favorite, and this will be asked about. 3 general concepts here. One, what is CRISPR-Cas9 used for? Bottom line, gene editing, really specific gene editing. CRISPR-Cas9, the way this works, let's see, there's usually a nice picture here. The way this works, CRISPR-Cas9, there's a special protein complex called CRISPR-Cas9. This thing, you can artificially give it a kind of a tracker sequence, okay? And it's a guide RNA, a guide RNA. You can give that to this protein complex, and what this protein will do is it will go and look everywhere in a target genome for a DNA sequence that is complementary to this thing. When it finds a region in the genome that it is complementary to, this CRISPR-Cas9 protein will cut DNA like so, which honestly is the hardest part in gene editing, is to specifically cut open DNA. Once you cut open DNA, you can use any other set of tools that we don't need to go over in detail to insert some new DNA into that opening. So that's what this is illustrating. CRISPR-Cas9 is done, its job is to cut open DNA at a really specific spot, and then you can insert new DNA in there, and you can edit the genomes in any organism with this system. That's why it's a very revolutionary system. Now, that's the mechanism, and that's the general function of it, is gene editing. As for specific details, I believe that is listed right here. So they give you some vocab words like the PAM sequence, and you may or may not be asked, if you're given a picture like this, and all the labels are removed, where is the PAM sequence? Where is the guide RNA in this picture? So it's all labeled here for you. So practice being able to identify the positions of those different vocab terms in this picture here. All right? That's the other important part with the CRISPR-Cas9 system. The first part, really specific gene editing. The second part, be able to label something like this for 1 or 2 questions. All right? Yes?
Just out of theory, would you ever ask what makes CRISPR more successful in finding sequences?
There may be at least an answer choice or something, and it has to do with the mechanism that I'm talking about. You're able to use a guide RNA for it to specifically find the specific target that you want to cut. In fact, for conceptual reasons, contrast this with the old way that we tried to do gene editing, and that is with restriction enzymes. With restriction enzymes, they go to areas in the genome that they want to go to, not where we want them to go to. So it's a little bit less specific. But for CRISPR-Cas9, we can design the guide RNA artificially and have it go to exactly where in the genome we want it to go to. There's another question over there, yeah?
You don't need to know certain sex?
No, retrotransposons you're not going to need to know for this exam. You may need it for the final exam. All right. Okay, oh wait, let me make sure. Did I forget something at the end of this? Yes, more HIV stuff. This is more of a video diagram thing, so don't worry about it. The conceptual information about HIV is already in the PowerPoints described earlier. So, 29, before we do that, let me check. Okay, so 29A. 29A talks about prokaryotic transcription. All right, so it's another section in your notes. They start by reintroducing to you the different types of RNA that you have: mRNA, tRNA, and rRNA. The interesting thing with prokaryotic transcription is a single enzyme is able to transcribe all 3 forms of RNA. That one enzyme is simply called RNA polymerase. Okay? There is no Roman numeral attached to the end of this, so very often people get confused with DNA polymerase Roman numeral 1, 3, and RNA polymerase here, and then there's DNA polymerase with Greek letters in them. So make sure you are writing these on separate sheets of paper and you're able to go over them. Since we are over time, I will not be able to write these things right now. But basically, one single RNA polymerase is able to transcribe all the forms of RNA that you have in prokaryotes. It is made of different subunits, each subunit having a known function associated with it. And in terms of transcription, prokaryotic transcription, it can be divided into 3 steps. Before we get to the 3 steps, we need to cover a concept called consensus sequences. All right. So, the way transcription works in prokaryotes and eukaryotes is that right next to the gene that you want to transcribe is a region called the promoter. In this promoter, there's certain patterns of DNA called consensus sequences. At each consensus sequence, DNA binding proteins will bind to in order to initiate transcription. In eukaryotes, we call those DNA binding proteins transcription factors. In prokaryotes, we kind of oversimplify it and just say RNA, say that the sigma subunit of RNA polymerase will bind to the consensus sequences in prokaryotes. So that's all you need to know in terms of prokaryotic binding, is that the sigma subunit of RNA polymerase is looking for a consensus sequence to bind to. I believe it is looking for the -35 region consensus sequence, and DNA will open up at the PribNow box. So, oh yeah, here's the vocab term, PribNow box, which is the consensus sequence that you find in prokaryotes. And here's the reason why it's often confused with TATA box. Look at the pattern of the PribNow box. That's why it is very often confused with TATA box in eukaryotes, yes?
So DNA is originally attached, but it scans and then it stops at the -35, right? Once it recognizes it?
Yeah, so the scanning thing, it's more like it's binding and dissociating from a bunch of random areas in DNA. So yeah, you can think of it as scanning.
Does it bind with low affinity at first?
I'll get to that when we get to initiation in a second. So, before we get to the steps, this is very, very important. You will get 1 or 2 questions on this following concept here, is consensus sequences. You may be given multiple different strands of DNA, multiple different promoters, and you may be given a consensus sequence. And you may be asked, which one is the strongest promoter? We talked about that at the very beginning. What's the first thing you need to look for to see if something's a strong promoter? It looks similar to the consensus sequence. The more exact it is to the consensus sequence, the stronger the promoter. Now, what if you have multiple answer choices that look exactly the same as the consensus sequence? You break the tie by looking at the other places around the consensus sequence. The ones with more A's and T's in it is better, because what's the purpose of these promoter sequences? It's where DNA is going to open up so that you can transcribe it. So, once you have your promoter, once you have your consensus sequence that looks really good, if you want to break some ties between consensus sequences, look at everywhere else. How many A's and